So something that we talk about a lot in anatomy is how structure relates back to function. And a lot of times when we hear structure, we think of, what does the shape of the bone look like? What does my eyes look like? What does my nose look like? But really, that structure goes all the way down to even the chemical level, right? So if I'm thinking about water, right, when I've got my oxygen and my two hydrogens, right, because we have we have our electrons here, they're a little negative, right? Got our little positives. On our hydrogens, right, we have that little kink in the structure, and that allows for lots of properties of water, which is a sort of different concept. But each chemical has a structure like this, which ultimately is going to change the structure of everything that it's built out of. So the big one that I wanted to talk about today was protein, right? So my protein structure, right, if we think about the different levels, initially I've got my primary structure, which has my amino acids, right? So my amino acids, right, those chemical amino acids. But each of these amino acids are going to have their own charges and their own shapes. Some of them are going to be bulky, some of them are going to be small, some of them are going to be positive, some of them are going to be negative. And that's going to change how it interacts with the environment, right? So as those changes occur, right, as we're responding to it, we have our secondary structure, right? So the main secondary structures that we think about, right? Let's just draw them kind of corkscrew through, right? We have things like my alpha helix or my beta pleated sheet. Which allow for, again, a little bit more complexity. But just because those are in that shape doesn't necessarily mean that, it's going to be able to do a lot, right? A bunch of corkscrews don't necessarily do a whole lot, right? A bunch of springs, right? If I just put a bunch of silly strings together, wouldn't necessarily make up something unique. And so these all fold together into my tertiary structure, where I have a mix of sheets and plates and random unstructured regions. And we're starting to see unique things like domains, okay, where those secondary structures interact with each other to allow for unique things to take place, right? Because think of it like this, right? One finger can't really do a whole lot, right? One secondary structure can't really do a whole lot. But as I add more fingers, right, so think more helices, and I've got a little plated sheet of the palm of my hand. Now these can interact with each other, right? So I can grab things, right? So these domains can do unique things. Sometimes we're going to have multiple strands together that can have their own quaternary structure, where we have a glob and a glob and a glob. And each of these have their own unique function that work together, right? And so in anatomy, we talk about some unique proteins, things like hemoglobin and myoglobin, that they change their shape, but that requires all the other parts of the protein to function the same. So as you're moving through, remember the shape of that protein, the amino acids that you find in that protein, right? The types of secondary structure you find in that protein, it's going to tell you a lot about what it does and how it's going to work in the body.